Alright, so right now, if you look at this given molecule, this is glycerol. So it has 3 carbon, 3 OH group, and rest are hydrogen around it. You learn triglyceride has glycerol right. This molecule is glycerol because it has 3 carbon and then 3 two. Okay, yes, already I mean, if you know the answer you can submit the answer, it's fine, right? uh, you learn starch, you learn glycogen, and you haven't learned albumin. You know DNA, it's normal basic molecules, everybody knows. Right, then you have a, you know that starch, we haven't seen such thing. Glycogen and DNA, if you know a little bit, they don't have this. Could be albumin or phospholipid. Okay, let's leave. We get this question again and we see the answer, right? Okay, soon we get the answer for this question. Some, I think this are already submit the answer. Now, when we talk about this fatty acid, it is also important to know there are 2 types of fatty acid. One is called saturated fatty acid. Other one is called unsaturated. Now, what is the difference saturated and unsaturated? So, saturated one, okay, just for the saving the time, I just only 3 carbon fatty acid is adding here. You can see it has maximum number of hydrogen ion. But if you look this one, they have a double bond. Yes. If you look at this one, the unsaturated fat, the most important thing is a double bond, having double bond. As a result, the hydrogen number is less. So it has a less hydrogen number and it has a double bond. So these double bonds, maybe 1 double bond, 2 double bond, 3 double bond, it depends on the fatty acid. When they have a lot of carbon in unsaturated fat, they can have a different double bond. Let's say I try to draw this one like this. So each represent the carbon and hydrogen, and then 1 double bond like this, and maybe another double bond here. So this has 2 double bonds. Okay, then unsaturated fat, again we divide into cis and trans. So what is the difference cis and trans? The cis carbon double bond, they have a hydrogen this side and this side. Trans carbon double bond, hydrogen is this side and this side. The orientation of hydrogen is different. The hydrogen cis has hydrogen on the same side, trans has hydrogen opposite side. Right, another important thing, when they make this double bond, the double bond is strong, very strong, and then the fatty acid long chain is bending like this. This is the double bond and it is bending. So we can see this in the cis fatty acid. But trans fatty acid, even they make the double bond, we don't see a bend because of the orientation of hydrogen. So this bend, also called kink, like this. Right, I think you can add this. Okay, another important thing, if we also draw this structure, they are also linear, like this. So saturated fat, they have again a line structure, only a cis unsaturated has this bend. So if we heard about this saturated and unsaturated, which one is a healthy one? So if we consume fat, lipid as food from animal or plant, which one is the healthy one, saturated or unsaturated? The full form of cis. Cis, the word is cis unsaturated fat, trans unsaturated fat, right? The word is cis unsaturated fat, trans unsaturated fat. Right, I have an answer. There are two answers. One is unsaturated is healthy and there's a none. Well, if we consume fat, whatever excess amount, it's unhealthy, because fat is the most killing food in human. The number one killing disease in the world is diseases which is related to the heart and blood vessels. If you search what is the most diseases associated with death in human, it is a disease coming, number one is a cardiovascular, heart and blood vessel related disease, cardiovascular disease. Now in this cardiovascular disease, if I am correct, maybe around 18 million people per year, they die with the cardiovascular disease, 18 million per year. So this cardiovascular disease is basically because of the fat and other factors. So then of course fat we have to select carefully, but we have to eat fat because it's a part of the body. Then we try to avoid eating a too much saturated one, and the unsaturated one is healthy. This is the healthy fatty acids. Maybe you heard about omega-3 fatty acid like this, fish oil. They are more towards benefits to the health than saturated fat. Now why is that? We will learn this unit 5 when we are learning cardiovascular disease. We are going to learn diseases of the heart and blood vessels. It is more towards the answer is cholesterol. Cholesterol metabolism in the body. Unsaturated fat is more giving benefits for the cholesterol metabolism. Generally they can lower cholesterol. So you will see the mechanism, but it's not important here. They can lower cholesterol, and saturated fat can shift to increase the cholesterol level, because cholesterol is come as another fatty acid, steroid. So cholesterol is we can group as a steroid, another steroid, right? All right, so next one, triglyceride. So under triglyceride we learn saturated and unsaturated fatty acid and triglyceride molecule. Second is phospholipid. Right, we take the glycerol molecule again. One fatty acid join here, and the second fatty acid join here. I explained how condensation reaction and this ester bond formation. Then to the third one, third carbon, triglyceride. Usually they have a fatty acid. The fatty acid can be, they can be different, maybe not the same, like here the long one. They may have a double bond like this. They can change. There's a lot of variations. So there's no rule. Any kind of fatty acid can join here, any kind of fatty acid can join here, and any kind of fatty acid can join here. But when you're talking about the phospholipid, there is a small exception. Here the phosphate group is joined. Now what is phosphate group looks like? It has a like this. That's a phosphate group. Now the phosphate group trying to attach to this side, a third carbon. But there is a small problem, because these negative charges give a huge polarity to the phosphate group, and they don't have any charges, because all the atoms are well established to make the bonds. So they have no reactive groups. They have nothing. They cannot link with the water molecules. They can't make a hydrogen bond with water molecule. And this region is called hydrophobic. Phobic is fear. So they fear to the water molecules because they cannot make a bond. When the phosphate group join here, the phosphate group mostly push to the other side. So then this side we can see the phosphate group with some kind of a charge. So they can react with water molecule. We call it hydrophilic. So you will see this is very important property in making the cell membrane, because this molecule has phosphate group, it gives the reactivity with water molecule, hydrophilic, and the rest of the molecule does not react with water, hydrophobic. So this is the structure of the phospholipid, right? So generally we symbolize it like this: the head part and the tail part. The tail and head. Tail is hydrophobic, head is hydrophilic. So this is the iconic picture for the phospholipid. Right, we will learn a little bit more in the cell membrane. So if you have enough time to copy this down on the phospholipid, now we can find answer for that question. If you have any question, you can send it to me, right? Okay, let's see the answer for this question now. Which one of the following compounds on hydrolysis will yield the above compound? I think last time Disat sent the answer correctly. Yes, this is a glycerol molecule. So, which one of the following compounds on hydrolysis? Now, what is hydrolysis? So, if this is a glycerol molecule and this one is a fatty acid, this one is a fatty acid, and this one is a phosphate group. So, hydrolysis is a break in the bond. The meaning of hydrolysis is we just add water molecule to break the bond. So when we break the bonds, we can get phosphate group, glycerol, and 2 fatty acid. So then answer is a phospholipid. Okay, albumin is a protein. Albumin is a protein. Right. The last one is the steroid, right? So under steroid, cholesterol, estrogen, progesterone, and testosterone like hormones we can list. I think these molecules we're going to learn many times when we are learning different lessons. So we can see these molecules and how they are important in the functions. Next, we are moving to the protein. Right, protein has carbon, hydrogen, oxygen, in addition to that, nitrogen. Now that's important. Protein has nitrogen, and some proteins they have a sulfur. Some proteins they have a sulfur group, right? Let's see what are the function of proteins, because it has many functions. Just get this. Okay. Right. What is the functions of the proteins? We can remember in this order: hamster, right? So okay, can you try to send a few function of proteins? Yes. Yes, it has many functions. It is a hormone. The proteins are hormone apart from the steroid hormones like estrogen, progesterone, and testosterone. These are main important three hormones, steroid hormones. Rest of the hormones are proteins. Then activity of the cells. That means recognition one cell with another cell. Cell recognition. Recognizing one cell with another cells. Yes, to make the cell membrane, it's a structural component of the cell membrane, and signaling molecule. So I explained you a signaling molecule before, a chemical which can come and bind to the cell, or maybe they can go into the cell and they can communicate with the cells to make some effective functions. Yes, transporters and also enzymes. So we know that most of the proteins are enzymes, and cell act as a transporters. For example, if cell is like this and there are proteins molecules located on the cell membrane, and they can selectively take a molecule into the cells. This is one example of transporting. We will learn cell membrane. We're going to see its role in the cell membrane. Right, next we're going to learn the monomer of the proteins. Now, monomer of the protein is called amino acid. Amino acid has carbon in the middle and it has 4 bonds. Now, 3 groups attaching to this carbon. Here, H hydrogen, hydrogen. This group is nitrogen and 2 hydrogen group is called amine group. And here carboxylic group. COOH carboxylic group. And here hydrogen. Right, now there are 20 amino acids. There are 20 different amino acids. All 20 amino acids, these three parts are the same. What is the difference? Only this group is different from one amino acid to another amino acid. So we call it variable group. Only this R group is different from one amino acid to another amino acid. So this is the structure of amino acid. So it has a central carbon, an amine group, carboxylic group, and hydrogen. The R is different from one amino acid to another amino acid, and we have a 20 different amino acids. Okay, let's see now how amino acids join together to make a complex structure of proteins. So I'm going to delete these notes. So hope you have time to copy this one. Right, now let's take one amino acid. So I write here R1, they are different. Second amino acid, here. It's different because this group is the only changing group in amino acid. Now when two amino acids join together, it make again same way. This hydrogen and OH join, and it goes as a one water molecule. So it's a condensation, and the bond is called peptide bond. And the bond is formed between the COO of this one and NH of the other one. So this is the bond. So it creates between the carbon and this nitrogen. So likewise, many amino acids join together one after another to make a long chain. So it creates a huge chain. It depends on the type of proteins. The amino acid number is different. Let's say insulin is a protein. It has a different amino acid. Let's take enzyme, lipase, amylase. So they have a different amino acid. Let's take a hormone like FSH and LH. So they have a different amino acid. But the basic starting point is amino acid first join together to make a long chain, and then this order of amino acid also different. Different amino acid, they have a different order to make a long chain. So you can copy this down under the monomer and making the polypeptide chain. And in the revision, the notes section, all these informations are given. And there's another word in the notes. So if this hydrogen ion left, this then it can make a negative charge. So this end is quite acidic. So it has acidic property in one end, and then if another hydrogen join here to this end, this gets positive charge. This positive charge represents alkaline nature. So then protein, because of these two different groups, they can have both alkaline and acidic nature. So such molecules are called amphoteric. So it has both acidic and alkaline nature. Right, now next thing is, initially it can make a long chain like this. This is just a starting point, but the protein is, after that they get different shapes. It is not a final version. To get the real property of protein, it has to change the shape. So this changing the shape of the protein, based on the changing the shape of the protein, we have several levels. We can categorize protein into several levels: first level, second level, third level, and fourth level. So the first level is called primary, second is called secondary, third one is called tertiary, fourth one is called quaternary. Then we have four different level of proteins. So let's see what are the four different levels and why they are important, right? Right, there is a question. We do this question before we move into the next. Right, you heard about this, the word anabolic. So anabolic together with catabolic, it make a word metabolic. Metabolism is the all the reactions which take place in the cells. So these metabolic reactions we can categorize into anabolic when it making molecules, and catabolic is when it's breaking the molecules. Can you send answer for this question? Yes, I think most of them correctly identify. Okay, anabolic is it's making the molecules. Now, starch is a polymer of glucose. So, starch hydrolyzed to glucose is breaking. Glucose is a building block of starch. Protein is the polymer of amino acid. Now, what is this word dipeptide? If there's a two peptide joined together, two amino acid joined together, then we call dipeptide. If there's many amino acids joined together, this is called a polypeptide. So dipeptide and a polypeptide. So it's a protein hydrolyzed to make a small unit dipeptide. Today we learn amino acid can make the proteins. It's making the proteins. So it's an anabolic reactions. And the conversion of fats to fatty acid, today we learn fat is made out of glycerol and fatty acid. So again it is breaking. So this is a reaction glucose to carbon dioxide and water. Respiration reaction, again glucose break down to carbon dioxide and water. It's again breaking, catabolic. Right, we have to join again for the next session after few seconds. Then we continue the structure of proteins and the levels of proteins.